Welcome, dear students. Today we are going to learn about immunohistochemistry markers in pancreatic cancer. So there are various types of pancreatic cancer. For example, we have pancreatic ductal adenocarcinoma, colloid carcinoma, medullary carcinoma, acinar cell carcinoma, PNET or PANNET, that is pancreatic neuroendocrine tumor, pseudopapillary tumor or pseudopapillary neoplasm, that is SPN, pancreatoblastoma, and some other rare type of tumors and metastases. These all are solid tumors. On the other hand, we have cystic tumors, which are serous cyst adenoma, which is benign type, mucinous cystic neoplasm, intraductal papillary mucinous neoplasm, and some cystic type of ACC, PNET, and SPT. All these pancreatic cancer have different type of IHC markers through which we can diagnose the exact type of pancreatic cancer and differentiate them from each other. So these are the various IHC marker for different type of pancreatic cancer. And we will talk about them in next slides. So first type we have is pancreatic ductal adenocarcinoma. We call it PDAC. So this is the gross appearance of PDAC. Here you can see is the tumor, which is yellowish white portion, poorly demarcated, surrounded by normal pancreatic parenchyma, demarcated by P. And here is the portion of normal duodenum. It is a cut section. And microscopically stained by H&E staining, we can see this small round granular structure, which are the malignant pancreatic glands and surrounded by dense pinkish stroma. So for PDAC, we have lots of IHC markers, and we'll start with TP53 and SMAD4. So in pancreatic cancer, we have a gain mutation of TP53 and loss of SMAD4 gene. So here in C, we can see this brown staining. This glandular structure is stained brown with TP53 IHC marker, which is shown by this arrow. And here, this structure is negatively stained with TP53 shown by arrowhead, which is a normal pancreatic gland. On the other side, in D, we can see the same glandular structure, which are the malignant pancreatic gland. They are negatively stained with SMAD4, and the normal pancreatic parenchyma or normal glands, they are stained positively with SMAD4. Another IHC markers we have are MAPSIN and PVHL. So this slide shows dual staining. One is brown stain and another is pink stain, pink or reddish stain. So these are the malignant glands which are positively stained with MASPIN, and these are the normal pancreatic acinar cells. They are stained positive for PVHL. The malignant glands are negatively stained with PVHL, and the normal pancreatic cells positively stained with PVHL, but the malignant glands are positively stained with MASP. Another staining we have is S100 and IMP3. So here on the left side, you can see this strong positivity for S100, and here you can see the cytoplasmic staining with IMP3, and you can see the blue nucleus which are negatively stained. So this is the cytoplasmic stain. And another markers we have for PDAC is MUC5AC. Here also you can see the strong cytoplasmic positivity, but the nucleus, which is in blue color, is negative. And here the malignant glands are negative for SMAD4, but these structures, the brownly stained structures, are positive. They are the normal pancreatic parenchyma. Another type of pancreatic cancer we have are colloid and medullary carcinoma. This is the gross appearance. Here you can see this variegated appearance, the fleshy, gelatinous appearance of the tumor, which is well demarcated round tumor surrounded by this pancreatic parenchyma. This is the head of the pancreas. This is the H&E appearance, microscopically colloid carcinoma. It has a large pool of mucin and the scanty malignant cells, glandular structures of pancreatic ductal epithelial cells. And on the other hand, in medullary carcinoma, we have a sheet of ductal malignant cells. It is called syncytial appearance. Medullary carcinoma and colloid carcinoma, they have peculiar IHC marker, which is CDX2, which is particularly positive in colloid carcinoma. You can see the positive nuclear staining, but it is negative in medullary carcinoma. So this is very useful IHC marker to differentiate between medullary and colloid carcinoma in pancreas. And the medullary carcinoma, it is positive for MLH1. Here you can see the nuclear, so though it is not strongly positive, but still it is positive for MLH1, shows nuclear positivity, but MSH2 and MSH6, they are negative in medullary carcinoma. Third type of pancreatic carcinoma, we have acinar cell carcinoma, ACC. So grossly, you can see this whitish tumor surrounded by some hemorrhage, this reddish portion, and this normal pancreatic parenchyma and some fat. Microscopically, we can see the cells have characteristic granular cytoplasm and small round to oval nuclei forming small acinar structures. This ACC is particularly positive for BCL10 gene. So here you can see strong positivity, which is shown by the green arrow. You can see the strong positivity for BCL10. But here we can see that these malignant cells or glandular structures are negative for BCL10 because they are not acinar structure. They are PANNET, pancreatic neuroendocrine tumors. So these are the neuroendocrine malignant cells. So they are negative for BCL10. But this here we can see the brown staining. So they are normal acinar cells, and that's why they are positive for BCL10. So BCL10 is very useful to differentiate between acinar cell carcinoma and pancreatic neuroendocrine tumor. ACC is also positive for trypsin. Here you can see these small dots of brown staining, and it is also positive for synaptophysin. Another type we have is pancreatic neuroendocrine tumor, that is PANNET. This is the gross appearance, yellowish white, yellowish flashy tumor. Here we have two foci, 1 and 2. They are well demarcated, round, fleshy tumor. Microscopically, we have trabecular architecture and pale pink cytoplasm along with eccentrically located nuclei. PANNET is strongly positive for synaptophysin. You can see the strong positivity, the diffuse positivity for synaptophysin. And here we can see moderate positivity for chromogranin. Another carcinoma is Another type of carcinoma we have is solid pseudopapillary neoplasm, that is called SPN. Grossly, it has variegated appearance, having solid and cystic area. You can see the small cyst along with some necrosis. The blackish area is necrosis. And microscopically, we have a solid nest of poorly cohesive cells forming a cuff around these blood vessels. So we have a pseudopapillary structure. That's why it is called solid pseudopapillary neoplasm. And SPN is strongly positive for beta-catenin. So here in the tumor, we can see this green arrow shows strong nuclear positivity. But on the other hand, the surrounding normal pancreatic parenchyma is positive for, like it has membranous positivity with beta-catenin, which is shown by this red arrows. SPN is also positive for CD10. Here you can see this small dot, tiny dots of brown staining surrounding the malignant glands. So it is CD10 positive, and it is strong positive for vimentin. Another carcinoma, so it is not carcinoma, sorry, another type of pancreatic tumor we have is serous cyst adenoma, which is a benign type. This is the gross appearance, and you can see the small multiple cystic structures which is gelatinous, and they are separated by fibrous, this white or yellowish fibrous septa. And microscopically, we can see innumerable number of tiny sub-millimeter size of cystic structures and surrounded by this fibrotic stroma. SCA is positive as it is, as I already said, it is a benign type of tumor, so it is positive for PVHL. Here we can see the tubular lining cells, the serous cells lining the tubular structures are positive, strongly positive for PVHL. And similarly, these tubular structure are not that much strongly like PVHL, but they are moderately positive for inhibin alpha and for MUC6. Next is mucinous cystic neoplasm. So grossly is it has gelatinous cut surface showing this ductular structures or cyst, and microscopically the cyst is very large, lined by intestinal or gastric foveolar type of epithelium. And the stroma is very characteristic. It is very dense and it is of ovarian type. So the stroma is ovarian type. And MCN is strongly positive for ER, estrogen receptor. Here you can see the brown staining, brown nucleus staining with ER. And the last type is intraductal papillary mucinous neoplasm. So grossly it has two types, and it is classified according to the involvement of the duct. If the main pancreatic duct is involved, it is called main duct type, and if it it has involvement of the branches from the main duct, then it is called branch type. So microscopically, it also has two types. One is intestinal and the other is gastric. So in intestinal type, we have long villous finger-like projections which are lined by columnar mucin-producing cells, and they resemble the villous adenoma of large intestine. That's why they are called intestinal type. On the other hand, the gastric one, they demonstrate gastric foveolar type differentiation with, here you can see the apical mucin and the eccentrically located nuclei. So these are the intestinal and gastric type of IPMN, and they are strongly positive for MUC6. So this is all about immunohistochemistry markers in pancreatic cancer. And thank you for watching this video. We will see you in the class for hands-on experience with microscopy. Thank you.